Hello friends, welcome back. In this video, we will discuss one more reading comprehension exercise that is asked for CAT and other management entrance examinations. Usual, the exercise is provided to you in the form of a PDF. The link to the exercise is available in the video description. So I would suggest that you please attempt the exercise first and then come back and watch the video so that you can clearly understand what were the gaps in your comprehension and how those gaps can be rectified, what were the mistakes you made in analyzing the questions, and how those can be rectified in your actual exam. So it will be more beneficial if you attempt the exercise first and then watch the video. All right. Before we go to the actual passage, this passage is actually a scientific passage. These kind of passages are generally asked in management entrance examinations because scientific passages are considered tough by the candidates. But there is an advantage as well as a disadvantage for scientific passages, if you ask me. The disadvantage is that since it talks about various scientific concepts, people may not be too comfortable reading and understanding those things. But once you read and understand it, the plus point or the advantage is that almost all the questions can be answered very easily because the answers are very specific. Since it is science, has got no gray areas where either it is correct or it is wrong. So it is very easy for you to analyze and answer the questions easily. So that you have to take advantage of that plus point by attempting the scientific passages. Don't get disillusioned or disheartened just by seeing a scientific passage. And most of the scientific passages or passages dealing with science that are given for management exams, they will be written in a layman's language. It may not be too technical. Yes, there will be some technical terms and all, but it will not be too technical. It will be written in such a way that any person with a reasonable level of English language and a reasonable understanding of science can easily read and understand the passage. So that is why you should always attempt these kind of passages. So here the number of words is around 444 words are there. So if you have a reading speed of around 200 words per minute, you would take around 2 minutes to read the passage. But one reading is not going to be enough for this particular passage or passages like this. You need to have a minimum of 2 readings plus the number of times you may have to come back to the passage for re-verifying your answers when you attempt the question. So if you are able to complete 2 readings within a matter of 5 minutes and then answer the questions in maybe taking around 1 minute per question. So there are 9 questions. So 9 plus 4, 13 minutes. You should be able to complete. In the exercise I have given you 18 minutes. So 5 minutes more I have given. So you can have 1.5 minutes for each question plus the 4 or 5 minutes you take for answering the or reading the passage. So this should be your strategy in this kind of scientific passages. So the first reading will give you an idea regarding the central idea of the passage and the really important points. The second reading will take you to the minute understandings, will help you understand all the complementary points or the minor details. So the 2 readings will give you a clear idea regarding the passage. So let us come to the passage. So what the passage is basically talking about, since you have attempted the question, you might be able to understand the passage is basically dealing with the principle behind the development of cells in an embryo, how these cells develop specific characteristics even at the embryonic stage. What is an embryo? If you have studied science, you know that embryo is the fetus or the smallest portion of an individual. When a male and a female mates, the female egg gets fertilized by the male sperm or the male whatever is going to fertilize the female egg. And once the fertilized egg or the egg is fertilized, it becomes an embryo, and the embryo develops into a new human, a new individual, or a new organism. So this is what happens. So here in this passage, the subject is how these cells in an embryo develop specific characteristics that indicate or that are displayed by the individual once that individual or organism matures. So how this cell determination, the concept or the term used is cell determination. Cell determination is nothing but how the cells of an embryo develop the specific characteristics that it displays at a later stage in life. So This was the question, and around 100 years ago itself, scientists or biologists had already found that in the case of invertebrate animal embryo. What is an invertebrate? Animals that do not have a vertebra or a spinal cord. Such animals, embryos, when they are cut into two at an early stage of an embryo development, these two embryos will develop. It is the embryo will not die. It will two embryos will develop into two different human beings. So the survival, that is when an embryo is in its very early stage of development, can be cut into two, and the two embryos will survive and develop into two different organisms or two different individuals of that particular species. And this led the scientists to believe that the cells in an embryo at an early stage are undetermined, means they have not yet developed any particular characteristics. That is why when they are cut into two, they develop into two different individuals or two different individuals, means members of that particular species. But the later biologists found that this process is not very simple. The survival of the embryo and the later development into two different individuals depend on the plane in which the embryo is cut. Means if you are cutting the embryo on a particular plane, similar to the plane in which the earlier scientists have done it, only then the embryos will develop into two different individuals. So that was the finding of the later biologists. Then what was the and the next question was how this was happening. So they understood that at an early stage of an embryo's development, if it is cut into two, it has the ability to develop into two different individuals under certain conditions, like say, depends on a plane and all those things. But how this is happening, that problem was still unanswered. What was the science? How these cells are remaining determined, or which of the cells are already having specific characteristics, which of the cells are not having specific characteristics. When do these cells develop these characteristics, and what were the factors that influence the development of these characteristics in embryo cells? That is it is explained by this term morphogenetic determinants. What is a morphogenetic determinant? Morpho means changing. Genetics means you know what is genetics. Determinant means factor. So factors that determine the genetic changes of an organism is the morphogenetic determinant. So what are the factors, or in simple terms, what are the factors that define the characteristics of the cells in an embryo? This was the question. This question remained unanswered for a long time. That then developments in molecular biology helped the scientists to answer some of the questions, or to develop certain clarity regarding the cell determination, or how the cells develop its own characteristics. And from this, they understood that this cell determination in an embryo happens even before the egg is fertilized. Means even before the embryo is born, in an unfertilized egg itself, the cell determination, the factors that influence the cell determination is already there. What is that factor? It is given in the third paragraph. The factor is a particular substance. That name of the substance is not given in the third paragraph. It is given only in the fourth paragraph. It is maternal messenger RNA. So this is the substance that is referred to in third paragraph. Contains substances. That substances are actually maternal messenger RNAs. But in the third paragraph, a scientist named Paul Gross, he did some research on sea urchins, an organism that is found in sea, and he found that these embryos have this particular substance, and it is not embryos, the eggs, unfertilized eggs have this particular substance, and which is a morphogenetic determinant, or a factor that determines the character of the cells in an embryo even before the embryo is formed. That is, in the egg, this particular substance is there, and this is found in the cytoplasm. So the cytoplasm is a particular area in the egg which is outside the nucleus, and the egg consists of, or the a particular egg cell consists of nucleus, cytoplasm, and everything is covered within a protoplasm. How do I know it? It is given in the second line. They are located in the cytoplasm of the egg cell. That part of the cell's protoplasm that lies outside the nucleus, means the cytoplasm is outside the nucleus but within the protoplasm. So how you can imagine the structure of an egg cell? Nucleus will be there, cytoplasm will be there. They will be separate, and both will be contained within the protoplasm. And this particular substance is in the cytoplasm. And this is inactive, means as long as the egg remains unfertilized, it is not active at all. Once, and it is not evenly distributed, it is unevenly distributed. It is not distributed homogeneously, means it is not distributed evenly. It is not uniformly distributed. It is ununiformly or non-uniformly distributed in the cytoplasm. And when the egg is fertilized, what happens? This particular substance or the morphogenetic determinant within the cytoplasm becomes active, and it interacts with the genetics or the genes that are coming in from the male counterpart of that particular reproductive system, and they they form certain characteristics for the cells in the embryo. So that is how this process takes place. And what is the how this morphogenetic determinant influences the cell determination in the embryos, because these determinants are called maternal messenger RNAs, and these contain, they are in the cytoplasm, we already know that. And once the egg is fertilized, these become active, we already know that. And once they become active, what they do is they influence the production of certain things called uh, histones. So histones are nothing but certain kind of proteins, and these proteins are developed with the help of this maternal messenger RNAs. And these histones, after they are produced, what they do, since they are these histones are produced by maternal messenger RNAs, so they will be in the cytoplasm because maternal messenger RNAs are in the cytoplasm. Once they are produced, they move into the nucleus. We know that nucleus is different from cytoplasm. So once the histones are produced, they move into the nucleus, and once within the nucleus, they get attached to the DNAs within the embryo cells. So once they become attached to the DNAs of the embryo cells, they form a bead and string kind of combination, because the DNAs will bind itself around the histones, and they form the bead, and then all these beads are connected together by other DNAs within the embryo, so that it becomes a bead and string kind of pattern. And this bead and string kind of pattern determines, or it decides the character or the characteristic of the embryo cell. Means this is how the cell determination happens. So once you understand this, what is the thing that we can very clearly say? Because the earlier scientists believed that the cells in the embryo were undetermined. It is saying in the first paragraph that they concluded that in the early embryo the cells are undetermined. But this study proves that even in the earlier stage, the cells in the embryo are determined with the action of the histones that are produced with the help of maternal messenger RNA. So this is the theory that has been proved with the help of advanced molecular biology. So this is the essence of the passage. So passage is basically talking about one discovery made by the earlier scientists and the conclusion they made out of it. And the later scientists proved that the discovery is correct, but it depends on certain factors. And the conclusion the earlier scientists made was wrong regarding the cell determination. The conclusion is that cell determination happens even before the embryos are formed, because the eggs already contain certain substances that determine the character of the uh, fertilized egg once the egg fertilizes. So that is why cell determination happens at the early stages of the embryo itself. And because when the embryo is cut into two, what is happening? Because of the uneven distribution of the these factors or this morphogenetic determinants in the cytoplasm, the action of these things on the cells of the embryo is very different. Because of the different actions, each cell in the embryo or displays they separate or distinct characteristics. That is why when the embryo is cut into two, it develops into two different individuals or two different embryos which have specific and distinct characteristics. So this is the understanding. So for your easy understanding, I have given a gist of the passage just after the passage is given. So if you want, you can just pause the video and go through the gist, because I have written it in a simple language so that you can understand the gist of the passage. Paragraph wise, I have given. So, but anyway, that we have already discussed the passage. Once the understanding becomes clear, the answer is very, very simple, or answering the questions will not take too much time. The first question, the passage is most probably directed at which kind of audience? So, what can you say about this passage? It is a passage that is directed to people who are basically studying about the development of embryos. What were the factors that influence the characteristics of the embryo cells, all those things. So, what can you say? Option A, state legislatures deciding about funding levels for a state-funded biological laboratory. Are we talking about funding? The passage or the paragraph passage is not at all talking about the funding required for this or any kind of money related issues. They are not talking. So this is definitely wrong. Option B, scientists specializing in molecular genetics. Probably correct, because this consists of molecular biology. We are not talking about molecular genetics, but we are talking about molecular biology and how it has helped in understanding certain concepts in genetics or embryonic science. So we can keep this option for the time being. Option C, readers of an alumni newsletter published by the college that Paul Gross attended. So this seems the alumni means former students. So a newsletter of a former organized by the former students, and the college which Paul Gross studied, they release the newsletter. So will a newsletter contain these kind of technical things? Normally, alumni newsletter consists of achievements made by certain people and some other things, contact numbers of the alumni, the old students, all those things. But here it is talking about the part, the purpose of the passage is not to talk about the achievements or what Paul Gross did. So the passage is mentioning Paul Gross just to highlight the way in which this theory was proved, or this particular invention was made by Paul Gross. It is not to talk about the achievements of the Paul Gross. So this probably is not going to be a suitable answer. We can ignore it. Option D, marine biology, studying the processes that give rise to new species. We are not at all talking about new species. We are only talking about how an embryo, when cut into two, develops into two embryos with distinct characteristics. So new species we are not talking about, and we are also not talking about marine biologists. We are only discussing about sea urchins, but the passage is also saying that so many other organisms were used in the research. It is not just sea urchins. The passage says that he and other biologists studying a wide variety of organisms. So it is not just sea urchins. Yes, he did some research on sea urchins, but he also did research on wide variety of organisms. So this cannot be a good answer. This may not be pertaining to marine biologists. Then undergraduate biology majors in a molecular biology course. Yes, we are talking about molecular biology. Yes, and undergraduate biology people will also be interested in embryonic science or the development of embryos. So this is a good answer. So you have two good answers: scientists specialize in molecular genetics, undergraduate biology majors. Which one do you think will be a better answer? See, when you talk about scientists, when a person is addressing scientists, he will not be using this layman's language. Most of the language will be technical language, because scientists anyway will understand the technical language, and the subject will be dealt at a very high technical level. But we can see that even we can read and understand this without too much of an effort. So these scientists may not be involved here, whereas undergraduate biology students means people who are all studying for undergraduation in biology. So they may not be having as much knowledge like scientists about this subject. So the language used will be slightly simpler. So this is why option D will be a better answer than option B. So we can go with option E as the best answer for this question. Okay, go to the second question. It can be inferred from the passage that the morphogenetic determinants present in the early embryos are. So, what is an inference? Any idea that is supported by a fact is called an inference. So, inferences are ideas that are supported by facts, or you have to need to have certain facts in the passage that supports the statement. So, you have to find such a statement. Option A, located in the morphogenetic determinants or the maternal messenger RNAs, where they are located in the nucleus of the embryo cells. Is it a correct inference? No, we know that these are not located in the nucleus. Where are they located? These maternal messenger RNAs or the substances morphogenetic determinants are they found certain substances which are which function as morphogenetic determinants. They are located in the cytoplasm of the egg cell uh, that lies outside the nucleus. So within the nucleus is wrong. So you can very clearly say that the located in the nucleus is wrong. Then option B, evenly distributed unless the embryo is not developing normally. Again, the passage says that these substances are unevenly distributed. Where it is given, not distributed homogeneously. In the unfertilized egg, these substances are inactive and not distributed homogeneously. So whether the embryo is developing normally or abnormally is not at all given in the passage. So this part is wrong. And evenly distributed again is wrong because the passage very clearly says that these morphogenetic determinants are unevenly distributed or not distributed homogeneously. Homogeneously means uniformly. So it is unevenly distributed. So not your answer. Option C, inactive until the embryo cells become irreversibly committed to their final function. What is this irreversibly committed to their final function means? They develop certain specific characteristics of their own, that is irreversibly committed. So these morphogenetic determinants inactive until the embryo cells become develop their own characteristics? No. Why? Because it is the morphogenetic determinants that help the embryo cells to develop the characteristics. They are inactive only till the egg is fertilized. Once the egg is fertilized, they become active. So the statement says that the embryo, these generated morphogenetic terms are inactive until the embryo cells develop their own specific characteristics. Is wrong because it is only with the help of these determinants that the embryo cells are developing their own specific characteristics. This is why this again is wrong. Option D, identical to those that were already present in the unfertilized egg, means the morphogenetic determinants are similar to those that were already present in the unfertilized egg, means the morphogenetic determinant is the early embryo. So, is this a correct statement or correct inference? We can see from this third para that the morphogenetic determinants are already present in the unfertilized egg. And once the egg fertilizes and becomes an embryo, they start action. So, identical to those that were already present in the unfertilized egg means what is the meaning of identical? Similar. So it means that they are similar to those that are already present in the unfertilized egg. What is the correct statement? They are the same. The same morphogenetic determinants that are present in the unfertilized egg, which are inactive, they become active once the egg is fertilized and becomes an embryo. So they are the same. They are not similar but same. So the key word is identical. Identical means similar, means you have a set of morphogenetical determinants in the egg and you have another set of similar morphogenetic determinants in the embryo. This is wrong because the passage says that the same morphogenetic determinants in the unfertilized egg are which were lying inactive, they become active once the egg becomes an embryo after fertilization. So word identical makes this statement wrong. Many of you will be tempted to go for this as an answer, but it is wrong because the word is identical. Identical is not correct. The correct word is same. So same as those that were already present in the unfertilized. Whereas identical means similar. Option E, present in larger quantities than is necessary for the development of a single individual. Means the morphogenetic determinants are present in the embryo in larger quantities so that it develops into a more than one individual, which is true, which is what the passage says that any embryo in its early stage of development, when it is cut into two, they develop into two different individuals. So what is the meaning? The meaning is that these substances are when the fertilized egg divides, the resulting cells are different from the start, so it can be qualitatively different in their own genetic activities. Means the cells are different because of the function of this morphogenetic determinants, and they are in abundance. Means even though they are not uniformly distributed, they are in abundance. That is why all the cells will form its own separate characteristics, so that this quantity is adequate enough to form two different individuals. That is why the last sentence in the third paragraph, fertilized egg divides, the resulting cells are different from the start. We know that. So the and so can be qualitative difference. So how this qualitative difference comes? Because the cells are having its own, all the cells develop its own specific characteristics from the start itself, from the beginning itself. They develop their own specific characteristic with the help of the morphogenetic determinant. So the morphogenetic determinant should be in ample quantity to develop this, right? So that is why present in larger quantities than is necessary for the development of a single individual. That is why different individuals are formed with the help of when the embryo is cut into two. So this is why this is an inference that we can clearly deduce from the facts. So anything that can be inference is something that can be deduced or something that can be understood from the facts given. So option E is the clear answer. Third question, the main topic of the passage. What is the main topic of the passage? It is about the development of embryos, or the development of specific characteristics of the cells in the embryo is the core concept of the passage. Very clearly, we can say that. So option A, the early development of embryos of lower marine organisms. Are we talking only about marine organisms? No, we are talking about invertebrates. Whether they are marine or not, we don't know. We have studied about one marine, or Paul Gross studied about one marine organism, which is a sea urchin, but he has also made this study on other variety of organisms, which is given in the fourth para. So we cannot say that it is only about marine organisms. So this is not a good answer. Option B, the main contribution of modern embryology to micromolecular biology. In the passage, is the embryology contributing to microbiology, molecular biology? No, it is the reverse, because of the developments in molecular biology, we have made some further understanding regarding embryology. This is given in the second paragraph. Recent discoveries in molecular biology, however, have opened up prospects for a resolution of the debate regarding the embryology. So that is why we can say that this it is actually the contribution of molecular biology to embryology. And is it the main contribution? We don't know. We only know that this molecular biology has contributed to the understanding of the modern embryology. Whether it is the main contribution of molecular biology, it is not given in the passage. So again, wrong. Option C, the role of molecular biology in disproving older theories of embryonic development. Is it disproving any older theory? No, it is the older theory was that when an embryo is cut into two, the two cut pieces can develop into two different embryos, which can develop into two different individuals. That was the older theory. And the conclusion from the older theory was wrong, but the theory itself was not wrong. So it is not the disproval of the older theory, and role of molecular biology disproving older theories on embryonic development. And the older theory was not about the development of embryos. It is about what was the theory that was actually disproved? It was about the cell determination. So the earlier theory said that cells in an embryo in the early stages of development, they were undetermined, whereas the new theory says that cells which in the early stages of an embryo, they are still determined from the beginning itself because of the function of morphogenetic determinants. That is why this embryonic development the theory is not wrong. The theory that is proved wrong is regarding the cell determination. The theory regarding embryonic development is still holding correct. So that is why molecular biology is not disproving that theory. Option D, cell determination as an issue in the study of embryonic development. Correct, because how cells are determined, how the characteristics of cells are formed when an embryo develops, whether cell determination happens later on or whether it starts in the beginning itself. All these things are clearly proven with the help of molecular biology. So this is the clear answer. But we can go for the last statement also. Scientific dogma as a factor in the recent debate. What is a dogma? Any statement that you are forced to believe without any questions raised. That is a dogma. Any statement or principle that you are forced to believe, or you are coerced, or you are commanded to believe something as true, you should are not supposed to question it. That is called a dogma. So whether any such dogma is there in this particular passage, something that you are forced to believe, no, that is wrong. And whether the debate is regarding the value of molecular biology, no, the passage is not debating over the value of molecular biology. So both the ideas are wrong. So very clearly, this option is wrong. So third one, D is the answer. All right, the next question. According to the passage, when the biologists believed that the cells in the early embryo were undetermined, they made which are the following mistakes? So this is talking about the wrong conclusion made by the scientists or the biologists in the early stages when they developed this theory. What was the wrong conclusion? They thought that in the embryo at the early stages of development, the cells were undetermined. That is why the cells, the embryo when it is divided, the cells are or the two different embryos are able to survive and able to develop into two distinct individuals. Whereas from the passage we can see that when the embryo is developed or formed, that at that stage itself the cells are determined with the action of morphogenetic determinants which are already present in the egg cell, and this action causes the cells to form its own characteristics. And because of the varying nature of the distribution of these morphogenetic determinants, the various cells display different characteristics. That is the understanding. So, which was the mistake made by the earlier scientists? That is the question. Option A, they did not attempt to replicate the original experiment of separating an embryo into two parts. Means the experiment was done only once. They did not uh, 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 do the experiment again and again. We don't know. So this, whether it is an assumption or a mistake, we are not at all aware because the passage is not saying anything about the whether replication is done or not. So you cannot think of this as a mistake made by the scientists. Option B, they did not realize that there was a connection between the issue of self-determination and the outcome of the separation agreement. Again, wrong. Why? Because the scientists understood that after the separation agreement, when the two embryos developed into two different organisms, they understood that it is because of the issue of cell determination. Means some cells were undetermined or something like that, they thought, where it is given in the second sentence. This led them to believe that the early cells in the embryo are undetermined. So the issue of cell determination was connected with the result of their experiment. They understood. So that is why they made this conclusion. Whereas this option says that they did not realize that there was a connection between the issue of cell determination and the outcome of the separation agreement. Wrong. They very clearly understood that this outcome of the experiment or the result of the experiment actually was because of the connection between the issue because of the issue of the cell determination. But how that was happening, they were not able to explain. That was the thing. All right, so this is not a mistake made by them. Option C, they assumed that the results of experiments on embryos did not depend on the particular animal species used for such experiments. Means what is the meaning of this particular statement? Scientists believed that this whatever experiments they did on embryos, it is irrespective of the animal. Any particular invertebrate, the same result will be produced. That is what they thought. Yes, correct. Well, this is a true assumption. This is not a mistake. Why? Because the earlier people, they did the experiment on invertebrate animal embryos. They did not do anything. They were not bothered about which particular animal they thought. So if they thought that depending on the animal, the outcome would change, what would have happened? They might have mentioned this here. But they were talking about all invertebrate animal embryos. So this is again wrong. Did not depend is wrong. Did not depend on the particular animal. This is their assumption. That is a correct assumption. So it is not a mistake because they assumed correctly that the results were independent of the animals on which they did the experiments, or whatever animals they invertebrates they cut the embryo, the same result was there. So they assumed that it is independent of the animal the species used. So this again is a correct assumption, not a mistake. Since we are asked to find a mistake, this is wrong. Option D, they assumed that it was crucial to perform the separation experiment at an early stage in the embryo's life. Again, it is a correct thing because the passage or the the initial discovery was that this division of the embryo has to be done at an early stage, only then it will develop into two embryos. This is not disproven later. So even at the later stages, it is said that only in the earlier stages of embryonic development, this separation or division of the embryos is possible. So this is again a correct assumption. It is not a mistake. The separation experiment has to be done at an early stage. So since it is a correct assumption, it is not our answer. Option E, they assumed that different methods of separating an embryo into two parts would be equivalent as far as the weight of the two parts was concerned. Means what? Whatever method you use in separating an embryo into two parts, the result would be same. Is it correct? No. The first para, last statement. Later, biologists found that it matters in which plane the embryo is cut. So it depends on the method you use to cut the embryo that determines whether it is going to be successful or not. If it is not, if it is cut in a plane different from the plane used by the early investigation, thus it will not form two whole embryos. Means the method is very, very important. So when you use the same method that is used by the earlier biologists, only then the development of two embryos will happen. So the method is very, very important. Whereas the statement says that they assume that different methods of separating an embryo into two parts would be equivalent as far as the fate of the two parts was concerned. This was the mistake made by the earlier scientists, which was found out by the later scientists. Because the later scientists found that the method which you use to cut the embryo into two is very, very important in determining whether the two parts will grow in as distinct organisms. So that is why this is a correct answer, because this is the wrong assumption made by the biologists or the earlier biologists. So this, we are talking only about the earlier biologists because it is the earlier biologists who said that the cells in the early embryo were undetermined. Later biologists found out that the cells in the early embryo also were determined. So we are only talking about the earlier scientists, which who are referred to in the first paragraphs. That is why option E will be the answer. 5. It can be inferred from the passage that the initial production of histones after an egg is fertilized takes place. So we know that the histones are produced with the help of maternal messenger RNAs, which are the morphogenetic determinants that are in the cytoplasm. So the cytoplasm consists of these morphogenetic determinants called maternal messenger RNAs. And once the egg is fertilized, they what they do? What they do once the egg is fertilized? They direct in large part the synthesis of the histones. What is synthesis? Production. Production in an artificial manner is known as synthesis. So histones are production under the influence of the maternal messenger RNAs. That is what we know. Where it is taking place in the cytoplasm? Correct, because where the maternal messenger RNAs or the morphogenetic determinants are situated in the cytoplasm, we know that. So initial production of histones also would be taking place in the cytoplasm, because the maternal messenger RNAs, which are directing the production of histones, are present in the cytoplasm. We know that. So option A is the clear answer. Still, for the sake of argument, we will go for the other option. In the maternal genes, no. The maternal messenger RNAs are produced by some of the maternal genes, but the production of the histones are not taking place in the maternal genes. So this is wrong. Throughout the protoplasm, again wrong, because the maternal messenger RNAs are found in the cytoplasm, whereas protoplasm is something that encompasses the cytoplasm as well as the nucleus. So throughout the protoplasm means the production of histones will be taking place inside the nucleus. Wrong, because the passage very clearly states that after the histones are formed, they go into the nucleus. So this is wrong. Option D, in the beaded portions of the DNA strings. Again wrong, because only after the histones are produced, they move into the nucleus. And once they are inside the nucleus, the DNA binds itself to them, forming the weirded portion. So this again is wrong. Now, option E, in certain sections of the cell nucleus. It is not at all in the cell nucleus. We know that cytoplasm is situated outside the cell nucleus. So how can the production of histones take place in the cell nucleus? Again, wrong. So very clearly, option A is the answer. See, this is how it how simple it gets. Once you understand the passage, you can very easily go through the questions without even referring to the passage again. Option 6, or question number 6 rather, it can be inferred from the passage that which of the following is dependent on the fertilization of an egg. So, what is dependent on the fertilization of an egg? What is dependent? We can very clearly say that the action of the morphogenetic determinants or the maternal messenger RNAs in producing histones is dependent on the fertilization of an egg. And the histones, once they are produced, they develop the characteristics of individual cells in the embryo. Option A, copying of maternal genes to produce maternal messenger RNAs. No, maternal messenger RNAs are the morphogenetic determinants. They are produced in the even before fertilization because they are present in the egg itself. So there is no need for fertilization to take place for this production of maternal messenger RNAs. Option B, synthesis of proteins called histones. Correct. Synthesis of proteins called histones are done only after the fertilization of the egg is taking place. So you can think of it as a probable answer. Division of a cell into its nucleus and the cytoplasm. How the cell gets divided into nucleus and cytoplasm is not at all stated in the passage. So this is wrong. Option D, determination of X cells potential for division. So again, the egg, whether the egg cells potential is not, egg cell is not dividing. It is the embryo cells that are dividing. So egg is unfertilized egg. So egg cells potential for division is, egg cell has no potential for division. It is the embryo that has a potential for division. And that happens only after the fertilization. So that is why this is wrong. Option E, generation of all of a cell's morphogenetic determinants means the cell's morphogenetic determinants are produced after fertilization. No, we know that the morphogenetic determinants are already present in the cell of an egg. So there is no need for fertilization for this to happen. So very clearly, the synthesis of proteins are happening after the fertilization of an egg. So option B will be the answer. According to the passage, the morphogenetic determinants present in the unfertilized egg cell are which of the following? Very clear answer. The last para, the substances that are found in the egg cells, which are the morphogenetic determinants, are called maternal messenger RNAs. These substances that Gross studied are maternal messenger RNAs. So, when Gross experiment was referred to in the third paragraph, these were only referred to as some substances. But in the fourth paragraph, the specific name of the substance is given. It is maternal messenger RNA. So very clear answer. You don't have to even look at the options. The morphogenetic determinants present in the unfertilized egg cell are maternal messenger RNAs. Option C. Then the next question. The passage says that which of the following plays a role in determining whether an embryo separated into two parts will develop as two normal embryos. So we know that an embryo in its early stage of development, when cut into two parts in a specific plane, then it will develop into two separate embryos. This is given in the passage. The stage in the embryo's life at which the separation occurs. Yes, this is a factor because it is only in the early stage of the embryo's life that this can be possible. So this is a true statement. Option D, see the instrument with which the separation is accomplished. The instrument is not at all mentioned in the passage. It says that in the plane in which it is cut. So it is not the instrument, the surface that is used for the separation is important. The instrument, whether the instrument, a specific instrument is required for the division, it is not given in the passage. So 2 is not given. Option 3, the plane in which the cut is made that separates the embryo. Very clearly that is given. So 1 and 3 are the statements that are the that indicate the conditions under which an embryo, when separated into two, will form two normal embryos. So 1 and 3. Option D. Question number 9. Which of the following circumstances is most comparable to the impasse biologists encountered while trying to resolve the debate about cell determination in the second paragraph? So, what was the impasse? Impasse means what? A stalemate, or they are things are not moving forward. You have hit a roadblock, or you have hit an obstacle which you are not able to surpass. That is an impasse. And this, what is the impasse that is situated or stated in the second paragraph? This is the impasse. What is that? The later biologists found that the discovery of the earlier biologists is true regarding the potential of an embryo to develop into two different embryos when it is cut into two at its early stage of development. But why this is happening, that they were not able to find. Whether which embryo cells had already its own characteristics, which when they become irreversibly committed to their fates, means when do the embryo cells develop its own characteristics, when do they become completely independent or completely committed to its own fates, fully develop its own characteristics, and what are the factors that determine a cell to develop its characteristics. So these where the debate, or the scientists were not able to explain how this was happening, why an embryo in its early stage of development, when cut into two, why it is developing the two different embryos, what were the how the cells are getting its own characteristics, when does the cell, a particular cell in an embryo gets its own characteristic, and what are the factors that influence the development of the characteristics of an embryo cell. These were the problems. So this was the impasse. So now you have to find a similar situation. Option A, the problems faced by a literary scholar who wishes to use original source materials that are written in an unfamiliar foreign language. Means what? The scholar does not know the language and he has a lot of problems in using the original source materials. Here, the situation is that we know that there is a theory, we know that this theory is correct, but we don't know how or why. Why it is happening or how it is happening. This was the impasse. In this case, the literary scholar is not even familiar regarding the foreign language, and he wants to use that source material. So this may not be a similar situation. So we will keep it as a probable option for the time being. Option B, the situation of a mathematician in preparing proof of a theorem for publication detects a reasoning error in the proof. What is the meaning? Reasoning error means the in the proof means this proof itself is wrong. So when you are talking about a mathematician who finds who wants to prepare a proof of a theorem which is inherently wrong, whereas in our case, we are trying to find the reasons of a particular theory which is correct. We want to find why it is happening. The theory is correct, we know that, but why it is happening, we don't know. In this case, the theory itself is wrong, and you want to write a proof of that theory. So this is not a similar situation. Option C, the difficulties of a space engineer who has to design equipment to function in an environment in which it cannot be first be tested. So this here it is talking about a situation where you have to devise a theory that is still untested. Whereas in our case, we have a theory that is already tested and proven correct, but we are not able to understand why it is happening. So again, this is wrong. Not a similar situation. The predicament of a linguist trying to develop a theory of language acquisition when knowledge of the structure of the language itself is rudimentary at best. So predicament means a problem. A linguist trying to develop a theory of language means he wants to develop the theory regarding a language acquisition, how to learn that particular language, how to learn a particular language. He wants to develop a theory regarding learning that language when the structure, we don't know the structure of the language itself. Means you are not aware of the structure of the language, or your knowledge of the structure is rudimentary, means basic knowledge. You have a basic knowledge regarding the structure of the language, but you want to develop a theory regarding the how to learn the language. So it is just like you have a theory that is proven, but you don't know the base or the fundamentals or the concepts regarding the theory, or you don't know you only have a basic understanding regarding the theory. In our case, you don't know the nitty-gritty or the why it is happening. In the same case, here you have a simple or basic understanding regarding the structure of the language. You want to develop a theory regarding the language. Is it possible? Not possible. So this is a similar impasse in my opinion. So keep it as a probable answer. Option D, the dilemma confronting. What is the dilemma? Confusion. Confronting a foundation when the funds available to it are sufficient to support one of the two equally deserving scientific problems. So here, two equally deserving theories or projects are there. You have to choose between the two. This is not similar because in our case, we don't have any two competing theories. We have only one theory, but we don't know the reason behind the theory. So this we can very clearly ignore. So you have two possible answers. The problem of a literary scholar wishes to use original source material where you have absolutely no knowledge about that language. And you have another theory where you have some knowledge about the language, you have to develop a theory of language acquisition. So in our case, when you compare between the two, you can see that the option D is more similar to our case where we have some idea regarding the division of embryos and how it is how when they are divided they form two different embryos, but we don't know the structure or the we don't know the what is happening underneath. That is the problem. In this case, option A, we are completely unfamiliar about the theory. We are completely trying to decipher or we are trying to understand something about which we have absolutely no knowledge. Whereas in this case, we are trying to understand about something about which we have some basic knowledge. So option D will be the better answer because it is similar to our case where we have a basic understanding regarding a theory. We need to find the nitty gritty or we need to understand why it is happening. So option D is the best answer. This is the only question which you should ignore or we should skip when you are if this question comes into your actual exam, because this question needs a little bit of more thinking and a very clear understanding of the nuances of the passage and its objective. Unless you have that idea, it is better that you skip the ninth question. All other questions are very easy. You should be able to answer those questions, except in the case of this particular question where that identical is coming. All other questions are completely straightforward. You can very easily find out the answers. In second question, maybe you will have some doubt regarding option D, but other questions except 9 are very easy. You should be able to find answers without a problem. See, everything depends on your understanding. So once I have explained it to you and you have listened to the explanation carefully, you might have very clearly seen that the problem where or the issue in where you have gone wrong with the questions is very clearly with the understanding. If you approach any reading comprehension questions in CAT with a half-baked understanding or a partial understanding, most of the time you may not be coming out successful in answering the questions. That is why comprehension is the most difficult part. Once your comprehension skills are developed, the application of the ideas and the application of the logic is something simple that you can very easily develop. So work on your reading skills and work on your comprehension skills by attempting these kind of questions and listening to the explanations very carefully so that you can understand which are the areas you need to focus on. Thank you. Bye bye. If you have found the video to be useful, please like, share, and subscribe so that I get motivated to come out with further videos.